This is a production of Cornell University.
Thank you so much for that introduction, and thank you guys for inviting me. This is probably my first time, I think, giving a talk to a bunch of plant biologists, but some of my best friends are plant biologists, so I won't begrudge you at all. And I did check though when I first got an email inviting me that it was clear that I did not study plants. That said, my highest cited paper is with plant biologists on digitization, mostly of plants. So clearly I should have been a plant biologist and I would be doing better than I am. But that said, bats are really cute. So it is what it is. So my talk today is going to be kind of a general talk on the impact of digitization, perhaps as I see it and as others in vertebrate biology see it, as well as novel methods for visualizing morphology. So as Bill was saying, there's a lot of overlap, I guess, in the kind of worlds of how to get in and visualize very, very small things. We want to do that too within the vertebrate world. You guys have been doing it, I would say, for longer than we have on plants. So with that, I'm going to jump in and just kind of give a brief overview of the kinds of questions that I'm interested in and the ways that I use digitized data, both from museum collections and from things like CT scanners in order to address these questions. So the basic question that I am interested in asking is how morphological and ecological innovations lead to the radiation of taxa. And this is necessarily broad because within vertebrates, I pretty much will work on any vertebrate that is interesting for answering and addressing this particular question. Depending on who I'm talking to, I'm a mammologist, I'm a herpetologist, I'm a vertebrate biologist. I have a paper on sharks. And as I said, my highest cited paper is on plants, which some of my plant friends never let me forget. Some of the fields that I'm interested in are related to bat skull sensory system morphology and how that relates to dietary ecology. I'll talk about this more throughout this talk, but I'm also interested in visualizing internal structures such as the endocast of vaginas and creating virtual models of penises in order to study copulatory fit in different vertebrate groups and understand how different sexual selection hypotheses have led to either increased copulatory fit or decreased copulatory fit, but what you need for that are 3D models of these structures. I'm very interested in avian lung physiology and morphology across the bird tree, and I'll talk about this a little bit more. And then more recently, I've been working with digitized presence-absence records of a variety of different animals to study biogeographic trends in reference to climate change. So the kind of thing that ties all these together, other than my basic question up here, is how can we visualize all of these structures and then collect quantitative data from them to answer our questions that we're interested in? So as I walk through this, I want to just talk a little bit about digitization first. So digitization is necessary because there are millions and millions and millions of museum specimens that have been collected over the past 2 or 3 centuries that are held in natural history collections across the world. And that's really, really helpful. Those data are fantastic. We're never going to be able to get data from 200 years ago. So we need to have these collections in order to access those kinds of data. But that said, accessing these data is a problem if you are not in one of these natural history collections. And these are just a few examples of things that you can do with these kinds of data, things that I'm sure people in this room are doing, studying using herbarium specimens in order to study relationship between increasing CO2, temperature, basically phenological questions like that. You can spread that out a little bit more and study things like pollution, climate change, habitat change, et cetera. But as I said, these are all in museum collections. And that is an issue if you are, for example, a researcher that has trouble getting to museum collections, and that can be for a variety of reasons. Underfunded researchers from countries that do not have large museum collections going back hundreds of years might have to fly to a new country, get visas, and do these kinds of things just in order to access data that perhaps we take for granted. This is also a big issue for students who may not have the funds accessible to go in and collect these kinds of data. So what can we do? Well, what have we done? This is nothing new. Digitizing these data is a major front across all realms of biology. I'm going to talk mostly about vertebrates because this is the thing that I'm most familiar with. These figures here are from the OVERT project, which is the Open Vertebrate Project, which is an initiative to take all of our little museum specimen friends and digitize not only the data on their specimen cards, what their species is, when they were collected, what their mass was, these kinds of things, but then also translate these specimens into 3D digitized forms that can then be accessed through open source platforms such as MorphoSource and things like that. And this has been going on, I would say, for about the last 20 to 25 years. OVERT itself dates back almost 10 years now. And the main goal, it's starting to wrap up now, the main goal of this initiative was to scan 80% of all vertebrate genera. And they've been very successful at spreading out across many museum collections in order to do this. Digitized records, though, do have some intrinsic problems with them. So these are data from the Harvard Museum of Comparative Zoology, as well as the Harvard University Herbarium. And what we're looking at here are the number of loans on our y-axis over time for different collections. So for example, ichthyology, ornithology, vascular plants. And what we can see is, at least based on these data, it seems as though sometime between 2000 and 2010, people started taking loans out. Doesn't really make sense. People were taking loans out for a lot longer than that. But all of those loan records were kept in notebooks, right? And nobody wants to go back and look at these like old dried up notebooks from the 1940s to see how many loans there were. So if we're interested in the impacts of this digitization effort and try to understand how digitization has increased access to specimens, then we're kind of hamstrung by the fact that our actual loan records or our visitor records are not being digitized, even if our specimens are being digitized. So when we look at this in a little bit more detail, we can understand more about what's been happening since these loan records have started to be digitized, since about the year 2000. And it does seem like it's increased access to specimens. So here in the red here, we have images sent to researchers just by count. And this is in the Museum of Comparative Zoology at Harvard. So basically, there were no images being sent pre-2005, and those data have been people have gone back with those data and double checked, one or two. But around the time that these records started to come online, people were asking for images. It makes sense, and it's great. So it seems like digitization has had some impact on specimen access. The next initiative that I would argue we need to do is start using these digitized data that have been put up in all of these online repositories and start collecting data from those digitized records. So for example, if we take our specimen of this frog here and then digitize him in 3D here, this is a soft tissue scan, which I'll talk a little bit more about later on in the talk, then you can post this online on MorphoSource. Anyone can download it. We could all go on MorphoSource right now and stop listening to me and download this frog and look at it. It's very cool. And then we could take that digitized data and wherever we are, go back in and collect additional data from those digitized records. So here is this frog's nervous system. Here's its cardiovascular system. Here's its muscular system. And then we can ask new kinds of questions about that. And this is what we have termed this digitization 2.0. Basically, all of this digitization 1.0 that's been really great and fantastic and has increased our understanding of what is available in museums has to happen in museums. We have to take pictures in museums, we have to scan specimens in museums, type up in Excel sheets, little specimen cards in museums. But now that those data have been digitized, we can then take them and add value to them outside of the museum collections themselves. So this is just sort of a general table that's super boring to look at, but these kinds of things. So For example, taking specimen images would be part of something that has to happen in a museum unless you get a loan and take it outside, but it's still associated with this museum directly. Whereas messing around with contrast enhanced CT data and generating data from that can happen in my lab. I don't have to go to a museum in order to do that. And this digitization 1.0 and digitization 2.0 initiative has been really successful. So this is just as of a couple of days ago. On MorphoSource, which is one of these main repositories, of which there are several, there are over 100,000 different media files that you can download for many, many different kinds of species. In fact, I have an undergrad in my lab who's interested in questions about hummingbirds, and we're getting all of her data from MorphoSource. So we're not having to go and rescan things that have already been scanned. Super useful. Other advantages here are that you can take these things and generate 3D printed objects. So this is just stuff that I had on my desk when I was making this presentation, but all of these different skulls have been used in different outreach initiatives here. And then here is a bird lung that is really, really hard to visualize, as you will see later on in this talk. But it's a lot easier if you can hand someone a 3D cast of that lung. Okay, so then that takes us to the second component of this talk, and perhaps maybe the more interesting component if you're already very familiar with the way that digitization works and how wonderful it has been. Maybe I'm tooting my own horn a little bit because digitization is not my idea, but a lot of these things are, so maybe it's not more interesting actually. But I'm going to walk you through different ways of taking these digital data and visualizing them. So novel ways for visualizing morphology. So we're going to talk first about visualizing hard tissues, then visualizing soft tissues, and then visualizing internal spaces. And all of these things are necessary for me to answer the questions that I'm interested in. So here we have a bat. Actually, my postdoc just put this together a couple of days ago, and you can see that we've segmented out some of its arm bones here as well as its leg bones. So visualizing hard tissues is not super hard because we can just do it with CT scans. So what can we ask with these kinds of data? So here we're going to see a bat fly in and catch this moth right here. And it's a very capable bat, catches it between its legs in this uropatagial skin fold that it has between its legs, and it's going to do a little somersault when it does that. So bats are a very interesting group for understanding diet, not just because they catch insects in a super cool way, but because they have a huge range of different diets. So within a particular family of bats that I study quite extensively, this family Phylostomidae, neotropical leaf-nosed bats, we have bats that of course feed on insects like most bats do, but we also have fruit bats, nectar bats, vertebrate-eating bats, fish-eating bats, and then our friendly blood-eating bats as well as a number of other different specialists that are not on here. So if we're interested in how morphology and ecology co-evolve in order to adaptively radiate, then let's go back to what is an adaptive radiation. So Schluter in this landmark paper in 2000 defined this as the evolution of ecological and phenotypic diversity within a rapidly multiplying lineage. So this particular group of bats, I hope I've convinced you just from those varied diets, are a ecological, do have a lot of ecological diversification, which we can see. If we look at their skulls, then they also have quite a bit of phenotypic variation. So these are bat skulls in silhouette here. This is the rostrum here, and this is the back of the skull. And these are the sharp pointy bits you don't want to get bit by. And then in addition, if we look at our tree of bats, there are about 1,400 different species of bat, then this group here that has these increased speciation rates relative to background are the same group of bats that have diverged into all of these different dietary niches. So as a result here, I would argue that we have an exceptional example of what Schluter defined as an adaptive radiation, the evolution of ecological and phenotypic diversity within a rapidly multiplying lineage that has these high rates of speciation. So how do we get these data? We want to ask questions. We have a great group to do it with. Well, we have a bat skull here, and The way that I typically collect data on hard tissues is using a technique called geometric morphometrics, which some of you guys may be familiar with. Basically, you put coordinates on your skull. This can be done in 2D or in 3D. So imagine that this is a CT scan of a skull here rather than just a picture. And then you pick homologous points across that skull. And then you can get rid of your skull and you will still have those homologous points that then you can use as your morphological data that are being input into your analysis. So as you come through here, we go to our museum, get our bat or get it off MorphoSource, CT scan it, and then generate our model for this landmarking method. If we take a look at some of the results that have come out of this work here, what we're looking at in this orange blob are our outgroups, so our non-neotropical leaf-nosed bats. And then along this axis here, we have a range of different diets of these neotropical leaf-nosed bats, where we have our nectarivores. I forgot that I did this, a nice little cartoon. Our nectarivores up here that have elongated their rostra, and then on the other end of that axis, these fig-eating frugivores that have shortened their rostra in order to increase their biomechanical advantage when they are biting into these giant fruits that are twice the size of their head. So it seems like this is an axis that is very informative for understanding what's happening with respect to this radiation of bat morphology. So what we were interested in next was asking whether or not these phyllostomid bats were evolving due to relatively low integration between parts of their skulls. So to explain integration and evolvability, we have our fox and we have our jerboa here. So fox limbs, because this fox is quadrupedal, are going to have these relatively highly integrated limbs. So its hind limb and its forelimb are going to have to evolve together. If its hind limb gets longer, its forelimb is also going to have to get longer. So two things have to evolve. The jerboa has its forelimbs freed from its hind limb. So its hind limb can get relatively longer without changing the forelimb at all. So this would be a situation where we would say that our fox has relatively low evolvability of limb length, whereas our jerboa has relatively high evolvability. And we can look at this just in this cartoon here, where as structures become less and less integrated, they become more and more evolvable. So coming back to our bats, what we expected, and it's been shown that generally mammals have these two different separate modules, similar to our fox with its forelimb module and its hind limb module, within their skulls, where they have this rostral module and this caudal module. So we figured, okay, seems like the rostrum is getting longer, the rostrum is getting shorter, and it's doing this very quickly in evolutionary time. So maybe it's not having to change the back of its skull at all, and that this bat skull is particularly evolvable among bat species. So the way that you test this is you would run integration analyses. So this is a theoretical visualization of this, where we have our rostral structure on our x-axis and our caudal structure on our y-axis. This would be a situation where you have very high evolvability, where there's no relationship between these two structures, and the points are able to go out in every direction, so relatively high evolvability. This would be a situation in red here, where we have strong integration between the front of our skull and the back of our skull, and you're really constrained in morphospace to a single line because these two things have to evolve together. So we were hypothesizing that we were dealing with this situation over here. When we look at our data here, we have a line that is following this prediction right here. So what we have is a situation where these bat crania are relatively integrated, strongly integrated, and they are still able to seemingly have relatively high evolvability, granting them this rapid morphological change. So why might this be? We can see on the ends of our axes here that the highly integrated structures on this red line are expanding outside of the morphospace that these black dots are. So what we're expecting here, perhaps, is that we would have really extreme morphologies in cases where we have high amounts of integration. And then when we think about that and then look back at our bats, we really do have extreme morphologies. So this short-faced bat here basically has no snout. And this is a long-nosed bat. There are actually some species of bats that have snouts that go all the way out to here for getting deep into flowers for nectarivory. So it would appear that these bats have been very successful by following this line of least resistance here and just evolving up and down it rather than out in morphospace, suggesting that tight integration may not always be related to low evolvability. Maybe tight integration, if you're on a particular axis of evolutionary change, will enable you to rapidly speciate. Okay, so that's visualizing hard tissues, one of the things that I do probably most. Recently, I've been more interested in visualizing soft tissues in order to assess things like muscle. The way that you do this is you use different kinds of solutions. This is Lugol's iodine here, which is a popular one for visualizing muscles. You put your bats in little jars, fill them with some percent of Lugol's iodine, 2.5, and then you wait a couple of weeks and then CT scan. You can see lots and lots of internal structure. So what we were keen to do is, after looking at these bat skulls, understanding a little bit more about their sensory systems. So here we have 3 different diets of bats, and we have 3 different primary sensory systems. So we were hypothesizing that since these bats are all relatively small animals, and they have to be small in order to accommodate their volant capabilities, that maybe they are doing some trade-offs with respect to their sensory systems, where nectar bats are going to prioritize their visual systems to see bright flowers, and they'll prioritize their olfactory systems in order to find smelly flowers. And they're not going to do very much echolocation. They do echolocation, but they're not using echolocation in order to find their food. So they're going to prioritize these 2 systems to the detriment of their auditory system. We might expect the exact same thing within our fruit bats, looking for bright fruits and smelly fruits. And then we would expect our insectivorous bats to prioritize their auditory systems compared to these others in order to find their food. So how do we get at these data? Well, once we have stained and scanned all of our specimens, then we have a lot of soft tissue data that we can go through with. So this is Corollia brevicata, a fruit-eating bat. This is its skull. And what we're going to do is walk through a video of the CT scan going from the back of the skull up to the front. So you can see some of the structures here, and we're going to make a few stops on the way. So here is our first stop. We can see this animal's brain. You can actually identify different brain tissue. It's not showing up super well there. You can identify different muscles. So this is the temporalis muscle, the main jaw closing muscle. You can see the tongue. You can continue up a little bit further here. Oops, I went too far, but we'll stop here. Here we can see the olfactory bulbs of this bat. We can quantify the area of the nasal epithelium along the inside of its nose. You can go a little bit further. You can see the size of its eye lens, calculate the size of its eye globe, and then you can do this throughout the skull of the bat across many, many different species. Scanning them in this way is just as easy as taking a regular micro CT. You just have to dunk them in this iodine or a different solution. There are a couple in order to get these kinds of data. And then you go through and then you segment out those structures in order to get quantified volumes. So that might look something like this. So these are a few of the data points that we were interested in quantifying in order to look at our 3 sensory system structures, so visual, olfactory, and auditory. And then this is a relatively simple plot before I walk you through a harder plot. Over here, we have our fruit bat, nectar bats, and then on this end, we have our insectivorous bats. And what you can see here is relative volume of these different sensory systems. So eyes in red, nose in blue, and ears in orange. So our fruit bats and our nectar bats were really prioritizing their eyes and their noses relative to their ears, whereas our insectivorous bats were prioritizing their ears over their eyes and their nose. So they were doing basically exactly what we would expect. So if we look at that in a little bit more detail here, we made tons of these plots. This is work that was done by a PhD student, Ronnie Hall. So here we're looking at visual systems versus frugivory here. So if you are a darker bar, then you are a strict frugivore. If you are a whiter bar, then you are not a frugivore. If you have a bar that goes to the, where are you guys looking? Right of this line here, then you have increased visual systems. And if you're to the left of that line, then you have decreased visual systems. So you can see pretty much right away that our strict frugivores here have enhanced their visual systems quite a bit relative to our non-frugivores. And this may have been one of the keys to their success. So that there are a lot of these plots. I'm just going to show you 3 of them. Here we have olfactory systems versus insectivory here. And what we can see is that our strict insectivores were not prioritizing their olfactory systems. And then here we have auditory systems and nectarivory, and our nectarivorous bats were not prioritizing their auditory systems. So this actually followed our hypothesis quite well, which was nice after our integration hypothesis did not work out in the way that we expected, but there it is. And then lastly, I want to talk about visualizing internal spaces, because this is quite different from the other 2 things that I've talked about. Basically, if you're visualizing hard tissue or soft tissue, what you're doing is you're scanning something and then either coloring it in a 3D modeling program, or you're thresholding that thing in order to select a part of that model. But with visualizing internal spaces, like we are with this crocodilian lung here, what we're actually interested in are the black values that are in the center, and then also some of these gray values as well. So I've been working with a colleague doing this in alligators and birds. I'm going to talk just quickly about some of this work in birds. Unfortunately, in order to explain this, you have to learn a little bit about how birds breathe, which is a kind of complicated thing. So birds don't have a tidal lung like we do, where we breathe in and we breathe out. They have a fixed lung that stays in place throughout their breath cycle. And they're also doing gas exchange both on inspiration and expiration. So when I inspire, I'm doing gas exchange. And then when I expire, I'm not doing gas exchange. I'm just getting rid of stale air. But birds, since they are really doing metabolically expensive things like flight, they have evolved this system for taking in air both on inspiration and expiration. So here we have a very simplified bird lung here. And then this gray is a bolus of air, so a single breath, we'll say. And this is its first inspiration pattern. This up here is its actual gas exchanging lung, labeled parabronchi. These areas down here are air sacs that are going to fill and unfill throughout the bird's breathing cycle. You can kind of think of them as storage tanks of either used or unused air. So when the bird breathes in, this air is going to go up into these parabronchi, but it's also going to fill this caudal air sac here. When it breathes out, the air in that caudal air sac is going to then go up and then go through the lung as well. So gas exchange is happening here on inspiration and then again on expiration. And then it's a little complicated because it's going to breathe in again and it still has this air from its first breath still in its lung. It didn't get rid of it. So it's going to use these front air sacs to hold that breath while the second breath is coming in and filling up the caudal part of the lung. And then on its second expiration, that air is finally going to leave. So this is just a kind of nice view of this. The gas exchange is happening again here in this parabronchial region. You can see how the air sacs are all associated with this. Now, unfortunately, for people that are interested in studying bird lungs, birds do all of this kind of similarly, but the differences in morphology are quite different. There's a lot of diversity within bird lungs for accomplishing this very crazy system that does give them the capacity to take in air both on inspiration and expiration. So in order to get at this question, really the first thing you have to do is you have to generate accurate models of lungs. So this is just a phylogeny of vertebrates going up from amphibians to birds over here, just looking at kind of structural differences in lungs generally. And the questions that I'm currently asking with my colleague here is how unidirectional flow in these avian and crocodilian lungs are affecting their lung architecture. So what kinds of differences are there within these groups, and how does that work? We're also interested in how differences in flight style may impact these changes in morphology. So if you're a hyper aerial bird, like a hummingbird, for example, maybe you require lungs that are capable of doing this even more efficiently than, say, a sparrow. So scanning these things is kind of, as you may expect, a little bit different from scanning a dead animal in a CT scan. So here we have a live hawk that's being scanned. And when this hawk is scanned, we'll be able to get a glimpse of it during its breath cycle. It depends on where it is in its breath cycle as to what its lung will look like. And as a result of that, we use both these live scans and intubated dead scans here. So here we have a hawk, a zebra finch, and a hummingbird that have been intubated, and their lungs have been filled to capacity. And then we're able to see what their lungs look like when they're completely full. And then we use that in order to generate these 3D models. So we're interested in doing birds. These are crocodiles here. We've published on this pretty extensively now, looking at differences in crocodile models using things like computational fluid dynamics, as well as different measurements here, which you can take off of these internal models of these CT scans and answer questions about allometry between species. These kinds of things can be done just like you're dealing with external soft tissue or external hard tissue. And just to show you some of these major differences in birds, which is where our main interests are now, you can see the diversity in shape difference between, for instance, an ostrich over here and an African gray parrot, where the African gray parrot has a much higher complexity than the ostrich does, even though both of these animals are breathing in functionally the same way. So hopefully I'll have more to talk about with respect to that in about a year. We're putting in an NSF grant in next month, actually, to expand that bird project. But generally, I wanted to talk about digitization, as well as visualizing hard tissues, soft tissues, and internal spaces, as well as give you a little bit of a window into my mind. Hopefully not too big of a window so that you don't go crazy, but I'd be happy to answer any questions that you may have. Thank you. Yeah.
So thanks for a great talk. To what degree is artificial intelligence being used to interrogate these sort of data sets and build correlations that haven't been thought of yet?
Yeah, so not very much within vertebrate biology. It is building the models. We are using interpolations, which is, I guess, a form of artificial intelligence, but like CNNs and things like that haven't really played very much of a role in that. And I know from my plant biologist friends that you guys, I think, are ahead of us on this, and that there's a lot that can be done. It is a little bit difficult because when you're dealing with like highly 3D structures like this, my understanding is that the I'll change off of this so you guys don't have to deal with that GIF for forever. The complexity seems like it's still a little bit too high for getting results in any sort of real time, but yeah, it's definitely building in that direction.
Yeah.
Yeah.
Yeah, when you talk about fast evolution or rapid evolution, what time frame are you talking?
Yeah, so these phyllostomatids are about 20 million years old, and they've diversified into about, they're the third largest back group, I want to say about 250 species. So rapid over deep time. Yeah.
How do you take into consideration the difference between the lateral and the axial resolution when you're doing a 3D reconstruction?
So with respect to like the CT scans, so you mean like the distance between slices versus the
So the imaging function of the X-rays, the diffraction going in the axial direction versus the lateral direction.
Oh, I don't know. The CT techs handle all of that for me, so I'm not exactly sure.
Thanks.
Yeah, sorry.
Yeah, this is sort of an off the cuff question but I see you at LSU Health. It's not uncommon for vertebrate paleontologists or vertebrate biologists to teach human anatomy in medical schools.
Yes.
I assume you're in a way, I suppose you're doing that here at vet school.
Yeah, I would say that that's true. So,
but I'm wondering, is there endangerment for the training the numbers of animal anatomists, vertebrate anatomists? So I know my own kid was a physician, had human anatomy from a paleontologist, which isn't unusual. So I guess what I'm asking is there a parallel in the paucity of training vertebrate biologists in morphology and anatomical evolutionary sense, if you will, with the paucity of training of plant anatomists, which is a rarity these days, too rare. Just an awful cuff thing.
yeah, so I don't know anything about there being a rarity with plant anatomists, but that is too bad. Yeah, so I would say that vertebrate anatomists often get jobs teaching anatomy because doctors don't know anatomy. So we don't know medicine. So I guess we have our specialities. But yeah, so that is a very common path, I would say. And that is basically what my path was, without working on fossils too much, but same basic principle. Yeah, so we're fortunate in that respect that med schools and vet schools really require us for training their first years.
That's great.
Yeah.
I have another question.
Sure.
So I work with a museum in Patagonia, and they use a lot of scanners to scan the bones from outside, and then they rebuild the fossils and they rebuild the whole vertebrae or invertebrates, whatever. Did you apply any of those techniques?
So how long do I want to talk about this for? So yeah, my PhD was actually on fossils, and one of the issues with fossils is that being in the ground for long periods of time crushes the fossils in different ways, and that changes their true biologic shape quite a bit. So for example, if I have my coffee mug buried in the ground for 50 million years, then maybe it'll come out kind of squished and sheared and compressed in different ways. How do we reconstruct biologic shape from that taphonomic artifact? I think that's what you're asking, basically. Yeah.
Yeah.
So you CT scan your taphonomically distorted thing, and then there are every single paleontologist, it seems like, has a different way of dealing with this issue. So there's no consensus yet on ways to reconstruct it. I would say that maybe there never will be, because there's no good method for doing that. So some of these methods include symmetricization. So basically, if you have two asymmetric things because of crushing, we pull them back together so that they're symmetric again. That has issues because it seems like making things symmetric doesn't necessarily make them the true original biologic shape. Gets rid of the asymmetry, so it cleans up the plots. That there are at least 5 to 10 different ways that is done. And this is what my PhD is on, which is why I won't go into it too much. But another thing that they do is if you have broken bones, then you put them back together digitally. That seems like it's, done in many different ways too. And I would say basically every paleobiologist should consider the data that are being put into their analyses, like how crushed do they think that they are based on their expertise. They're the ones that work on these groups. How real do they think the trends are that are coming out of those analyses? But yeah, it is done a lot with paleontology.
Yeah, they do it constantly.
Yeah, this
And,
issue is
you
why I
can
don't work on
calculate
fossils
actually the pressure.
anymore.
Yeah, you
Actually.
can calculate the pressure of the sediments based on the amount of time that they were deposited and what type of deposits you have.
Which is what needs to be done and has not been done.
Of course not, there's no money to do that.
Yeah, it would be great.
It's too expensive.
Yeah. So it's a lot easier to just ignore the problem, which is also, I think, a method that many paleontologists will use. But I have strong opinions.
Oh, yeah.
Yeah.
So do you have your own scanner? Can I ask you to shamelessly plug the nano and micro CT imaging capacity we have at Cornell?
Yeah, so I can talk quite a bit about that because I just put in a major equipment grant for getting a new scanner. So we currently have 3 micro CT scanners on campus, as well as 2 CT scanners at the vet college that will get down to maybe 600 microns-ish. The 2 that we have, or we have 3, but there are really only 2 because we have 2 of the same one at the Biotech Resource Center. One of them will scan things that are really, honestly, almost exactly this size. It's made for scanning in vivo mice and rats. So it can be quite good if you're interested in sort of like a lower quality scan that is of a relatively larger object that is still small enough that it would fit in a chamber that's the size of my coffee mug. It goes down to about 10 microns. The nano CT that we have will scan anything, I think, about that would fit on this table, but it does it at an incredibly high resolution. So I think it gets down to, I want to say like 300 nanometers or so, and then it can scan up to about 30 microns. So that's the worst resolution that you get is 30 microns. The problem with that is it's scanned by the hour. The larger your object is here, and if you're scanning it at like 100 nanometers or something like that, it can take like 7 hours, and I think it's $110 an hour. So that's not necessarily the best. The one that we put in that hopefully we'll have in the fall, if this grant is successful, would scan larger things at lower resolutions, so sort of in between these 2 scanners, where you can scan something the size of this table at 10 microns in an hour, but you wouldn't be able to get down to the nanoscale. So there's your shameless plug. Hopefully that's good.
I want to thank Brandon for a very interesting talk. Remind the grad students and postdocs that there will be a lunch afterward now in G22, Plant Science. And again, thanks. Very interesting, very fun.
Thank you, guys.
This has been a production of Cornell University, on the web at cornell.edu.